So my disease and vector ecology laboratory is studying the black-legged tick, which transmits Lyme disease, because Lyme disease is the most commonly transmitted vector-borne disease in the United States, and it's the 7th most commonly reported notifiable disease in the United States.
This is ideal nymph habitat. So it's rained the last couple of days, so it's moist, it's cool, it's perfect conditions. We have a nice canopy providing shade, and the nymphs are found down in the leaf litter, and there's a lot of leaf litter in here, and they will come up on these club mosses and they'll quest and just wait for an animal. Could be a mouse, could be a person, could be a deer or whatever, to walk through here, and then they grab on, latch on, and then crawl up and find a spot to attach. Perfect, that's about 1 meter. Yeah. So during peak nymph activity, there's 3 nymphs for every 2 square meters. That is, I'm sharing this space with 3 nymphs.
The ticks are collected using a 1 square meter drag, which is made of a heavy muslin cloth, and you just drag it along the ground. We set up plots so that we know where to drag and we know the correct area to drag.
Even though Pennsylvania ranks so high, or ranks the highest in reported cases, until last year there was very little known about the distribution of infected ticks in Pennsylvania. So that's why we've decided this is a really important public health project or problem that needs to be addressed.
So this is the size of a nymphal tick. It's approximately the size of a poppy seed. So most importantly, we have nymphs so we can practice extracting the DNA and testing them for pathogens. So about 25% or 1/4 of these nymphs will test positive for the Lyme disease bacterium.
Some of the instruments that we specifically use in this project to detect tick DNA, to detect nymph DNA, and the spirochete nucleic acid molecules carried by the adult tick and the nymphs includes the real-time DNA thermocycler manufactured by Eppendorf. We also have the conventional PCR equipment called the Biorad DNA thermocycler. This allows us to amplify DNA molecules in a very conventional way, which is followed by gel electrophoresis and use of digital documentation system to look for those specific target nucleic acid molecules that are being amplified by conventional PCR methods.
So we've been conducting research on the black-legged tick and Lyme disease for the past 3 or 4 years in collaboration with the Pennsylvania Department of Environmental Protection, the Northeast Wildlife DNA Laboratory, the Allegheny County Health Department, University of Pittsburgh Medical Center, and the U.S. Forest Service.
So once the ticks actually make it to the lab, we sort them out into male and female groups, and then we count them all to make sure how many ticks we have in total. And then we have to make sure that they're all black-legged ticks. So we have a taxonomic key that we use to identify physical features of the tick, and that will tell us whether it's a black-legged tick or not.
And what we have found, we've done a statewide study with the Pennsylvania Department of Environmental Protection, and we found infected ticks in every county in Pennsylvania, from Philly to Erie. Previously, it was thought that the black-legged tick and Lyme disease was more of an eastern Pennsylvania problem, but it's really throughout Pennsylvania.
This is a very much a dedicated laboratory for microbial forensic research. This laboratory was established with tax dollars, so I'm very proud that my lab is able to collaborate with Dr. Simmons, 2 of his students, and students working in the field, and bring the field material to test in the laboratory to specifically analyze these biological agents, which are of great importance in Western Pennsylvania.
The first step of DNA extractions involves quartering adult ticks or having the nymphal ticks, and then they are placed in a 1.5 milliliter test tube where the lysis buffer is added overnight and the cells burst and the proteins are eaten away.
The Lyme disease bacterium is actually passed back and forth between hosts and ticks because out in nature there's a lot of hosts who are already infected from previous tick generations, and then new ticks will hatch and attach to those infected hosts. The host will infect those new ticks, and the ticks will go off to infect other hosts.
The third step of DNA extraction involves precipitating the DNA, which means making it solid, which is done by adding alcohol. The final step is eluting the DNA, which means that the DNA is washed through the filter, so then what's left inside the tube is just DNA.
PCR is polymerase chain reaction. This technique allows us to amplify small amounts of target DNA. First, the samples were loaded into the machine. Specific probes were used to target these nucleic acid molecules. The protocol involves denaturation by heating the samples to 95 degrees in 1 second. This cycle runs 60 times for each reaction. If the target nucleic acid is present, there is a steady increase in vitro amplification. The results are captured on the computer screen real time.
So it's thought that when the Europeans colonized North America, they deforested the landscape, and due to hunting pressures, they eliminated deer and other wildlife. And in fact, it's hard to believe, but around 1900, the turn of the century, the deer was extirpated and almost extinct. And when the forests were removed and the wildlife were removed, which are the hosts for the ticks, the tick disappeared as well, and so did Lyme disease. And it's believed that it survived in refugia along the northeast, Long Island and Nantucket and Martha's Vineyard and other coastal areas, and also survived along the coastal areas of the upper Great Lakes states along the Great Lakes. And since that time, the forest has come back, the landscape has been reforested, and the wildlife have come back, including the deer. And on top of that, or layered on top of that, we have a different type of forest now. We have a forest that's been fragmented in the Northeast by development, whether it be suburbia, shopping malls, or whatever. And in addition, we have climate change to deal with. We have invasive species, which can provide good habitat for ticks as well. So it's complicated, but it's believed that the tick is simply recolonizing places that it used to exist. So in partnership with our collaborators, Indiana University of Pennsylvania's Department of Biology is at the forefront of studying the black-legged tick and Lyme disease in Pennsylvania. For example, our contribution to the statewide survey and also the population biology of the black-legged tick in Western Pennsylvania, both of which were published in the Journal of Medical Entomology last year.
We try to dress appropriately. We wear really light colored clothing where you tuck everything in, tuck your shirt in, tuck your pants into your socks, and we wear high permethrin treated tube socks, and that's a pesticide and it'll kill the ticks. You always check for ticks after we come home, so shower right away and check all over and remove with tweezers if you find any.
Because our research is so important in terms of public health, outreach is a very important component, and we involve students in outreach activities and give workshops and presentations to community groups routinely. Also, we share this information with government agencies so that they can better manage tick-borne diseases in terms of surveillance, control, and prevention. And lastly, we publish our findings in journals such as the Journal of Medical Entomology, so that we're adding our findings to the scientific literature, that base of knowledge, so that others can benefit from it.
In many places like university, for example, you write a paper and you submit it and you hope for the best. Here, you don't do that. You just work every now and then, you show people, and then you work again. So it's not a matter of the final product as much as you have to show people what you're doing right now just to get feedback and then continue editing later on. To edit the videos for Lyme disease and other projects, we mainly use Premiere. However, we also use After Effects and Illustrator. And since I don't have the enough experience to use those, we actually have Paul, who works in After Effects, and Anthony, who actually works in Illustrator. And we all work together to make the project that you see at the end.
All right, for Lyme's disease, I was able to work with Dr. Simmons and Dr. Barathan to conduct infographics. So I took statistical information that was given to me and had graphs that were already made and were able to make them fit the video more and have its own identity. So I was able to create vectors of ticks. What a vector is a scalable image, so it could be used on anything. So I recreated ticks, graphs, and other statistical information to be used throughout the video.
And then what program did you use for that?
Oh, the programs that I used was basically just Illustrator. I worked with Paul on several different projects, but for the Lyme's disease project, we were going to create, take the ticks and animate them to give a little bit of more movement into the actual video process itself. But most of the animations me and Paul worked on were maps.
And then
for Lyme disease, I only did about 2 animations. I did, it's kind of just like a slow turning state map with all the counties and how bad Lyme disease is in each county, basically. And then I did the intro graphic, which was the cells dividing and the title. But basically, one new thing I learned from doing the title is working with 3D space with graphics. That's what the biggest and hardest challenge I kind of had with the first title intro is kind of maneuvering everything to make sense when the camera moves inside the software on each axis to know where it's going.